Hello to everyone. My name is Hussain, and today we'll be seeing part 5 of chapter 2 with biological classification. Part 5 includes your kingdom Mycota or kingdom fungi. Okay, under this topic, we will study the characteristics of fungi, the nutrition which a kingdom fungi has, and reproduction in kingdom fungi. Okay, so let us start with the introduction. Study of fungi, as we all know, is called mycology. Anything which has myco in it, mycota or myco, it has something to do with fungi. Okay, yes, so study of fungi is called mycology. Myco meaning fungi, logy means a study. Okay, P.A. Mitchell is known as the father and founder of mycology. P.A. Mitchell. Okay, this is the only one person which you have to keep in mind with respect to NEET. Okay, so just like bacteria, fungi are also cosmopolitan or ubiquitous. They are seen everywhere in the sea, in aquatic habitats as well as in terrestrial habitats. Okay, and all over the world. Okay, they generally grow in warm and humid places. And all fungi are heterotrophic in nutrition, meaning they are either saprophytic, parasitic, or symbiotic. They are either saprophytic, parasitic, or symbiotic. Fungi range from unicellular fungi such as yeast to multicellular complex fungi such as mushrooms. Okay. Yes, examples of such some fungi are bread mold, which is nothing but Albugo candida. We have orange rods, we have mushroom and toadstools, etc. So white spots on a mustard leaf are due to a parasitic fungi called Albugo candida, as seen in this picture. No, this is white spot on mustard leaf. It is caused by a fungi called Albugo candida. Okay, and then there are some fungi are sources of antibiotics. Example is penicillium. Okay, penicillium gives you penicillin. Okay, penicillin is the antibiotic which you take when you get infected by a bacteria, right? So you have penicillium, from which the penicillin is extracted. Okay, then we have unicellular fungi, as we discussed, called yeast. Okay, and other fungi which cause diseases in plants and animals. Okay, one such example is wheat rust by Puccinia graminis. Okay. Yes, coming to the next feature, which is except in yeast, fungi are filamentous. So the body of the fungi, when you say fungi, or when you say, let us say, when you say a dog or a cat, you imagine how a dog or a cat looks like. But when you talk about fungi, you are not able to imagine what it looks like. It is a fungi generally looks like threads. Okay, it is nothing but pieces of thread. Okay, so the thread is of two types. Okay, so since they are thread-like, they are called as filamentous. Okay, meaning the cells are arranged in a thread-like structure. Okay, the long, slender thread-like structure called hyphae. So a single thread-like structure, be it branched or unbranched, is called a hyphae. When many thread-like structures come together and form a network, then it's called a mycelium. Okay, then it's called a mycelium. Okay, singular thread-like structure is called a hyphae. Plural of it, when they come together and form a complex network of hyphae, then it is called as mycelium. Okay, now hyphae is of two types. Okay, a single cell. Okay, for example, if you see the second diagram here, when you see the second diagram here, you see that there is a single hyphae, there is a single cell, there is no partition between the cells. Okay, but this single cell has multiple nuclei. So when a single cell has a multiple nuclei, then such a cell is called a cenocytic cell. And since this is a hyphae, then the hyphae is called a cenocytic hyphae. It is nothing but it is a continuous tube filled with multinucleate cytoplasm. Okay, multinucleate cytoplasm. And the opposite of that is septate hyphae, where septa meaning wall, cross wall, or in this case it is cell wall. When cell wall is present between the cells and each cell has only one nucleus, then such a hyphae is called as septate hyphae. So in contrast to septate hyphae, cenocytic hyphae can also be referred to as aseptate hyphae. Okay, so speaking of septa, the cell wall of fungi is made up of chitin and polysaccharides. It is not made up of cellulose just like plants. It is not made up of peptidoglycan just like bacteria, as we saw in the previous classes. Here the cell wall is made up of a new component which is called as chitin and subsidiary polysaccharides. Okay. Yes. Now coming to the nutrition in kingdom fungi. as we all know that fungi are saprotrophs. Okay, mostly because they feed on or they absorb nutrition from dead and decaying organic matter or other soluble or other organic matter. Okay, so first one is saprotrophs or saprophytes. Okay, so saprophytes are of two types. One is obligate saprotrophs. Obligate saprotrophs are nothing but they are compulsorily saprotroph, meaning they are only saprotrophs. Okay, they do not have any other nutrition in their life cycle. Okay, so such saprotrophs are called obligate saprotroph, meaning compulsory saprotroph. Okay, they obtain food only from dead and decaying organic matter. Okay, only from that, nowhere else. Example is Rhizophus nigricans. Okay, second one is facultative parasites. Now, do not be confused with the name. Facultative parasites are saprotrophs only, which, when dead and decaying matter is not present, they feed on live matter as well. Okay, they become parasitic as well. Okay, so they are normally saprotroph, but in the absence of dead organic matter, they become parasitic. Okay, so such organisms are called as facultative parasites. Okay, yes. So we have obligate saprotrophs and facultative parasites, which are both saprotrophic in nutrition. Okay, example for facultative parasites is Rhizophus stolonifera. Okay, next are parasites. So some of the fungi are parasites as well. This parasitic fungi have a structure called as hostoria, which is a root-like structure which is embedded into the cells of the host, and it absorbs the nutrition and organic matter from the host. Okay, such a structure is called hostoria. It can be understood as synonymous to roots. Okay, hostoria, which helps in the absorption of nutrition or nutrients from the host. Okay, the hyphae of the fungus help to penetrate into the body of the host using mechanical force and chemical force. Mechanical force is through push only, and by softening the cells with the help of chemicals, which is nothing but enzymes. Okay, secreted by the hyphae, which is the fungi. Okay, so parasitic fungi are again of two types. One is obligate parasite, meaning they are compulsorily parasites only. So they fail to survive in the absence of the host. When the host is not there, they die. Example is albigo candida. Okay, the second one is facultative saprophytes, meaning that they are parasitic when the host is present, and when the host is not present, they become saprotrophs. Okay, or saprophytes. Okay, so they are normally parasitic, but in the absence of a host, they become saprophytic. Okay, yes. And the third type of nutrition is your symbionts. Okay, symbionts include lichens and mycorrhiza. Lichens is nothing but a symbiotic relationship between algae and fungi, where the fungi part of the lichen is called mycobiont or fungal part, and the algal part is called as phycobiont, since phyco relates to algae and myco relates to fungi. So phycobiont plus mycobiont come in close proximity to each other to form a new organism called lichen. So since two organisms are involved, these lichens are also known as dual organisms. Okay, they are of different types. We have crustose, which are paper-like. We have foliose, okay, which is leaf-like, and then we have fruticose, okay, which is branched. Okay, and the second type of symbiotic relationship which is seen in fungi is mycorrhiza. Okay, mycorrhiza is nothing but a symbiotic relationship between fungi and the roots of higher plants. Okay, roots of higher plants. There are two types of mycorrhiza seen. Ectomycorrhiza, which penetrates into the host, but does not penetrate the cell. That is why it is called as ectomycorrhiza. And the other one is endomycorrhiza, which penetrates into the cell and forms vesicle, sac-like vesicle, and tube-like arbuscle. Okay, so that is why endomycorrhiza, also known as VAN, vesicular arbuscular fungi. Okay, yes. So next, let us come to reproduction. Reproduction is of three types. We have vegetative propagation, we have asexual reproduction by spores, and we have sexual reproduction by sexual spores. Okay, so starting with vegetative propagation. Vegetative propagation happens by three types. Okay, fragmentation, where when the hyphae breaks into different fragments or a few fragments. Each fragment develops into a new hyphae. Okay, a new hyphae. Such a phenomenon is called as fragmentation. Next is fission, your simple breaking up of cell into two. Okay, where first the nucleus breaks and after that only your cytoplasm breaks into two parts. Okay, into two cells. And the third one is budding. Okay, where a bud arises from the cell, from the mother cell, and it buds out into forming a new cell. Also, when the wherever the new bud is formed, on the parent, there is a scar that a budding has taken place. So with the help of the scar only, you can determine the age of the organism. In this case, it is yeast. Okay, so you can determine the age of the yeast, how old is the yeast, depending on the number of birth scars there are on the yeast. Okay, then the second reproduction is asexual reproduction. This takes place with the help of asexual spores such as conidia, sporangiospores and zoospores. Here the conidia or the conidiospores is your ectomorphic spores, or they are made outside the cell. Okay, outside ectogenous spores, where they are made outside a structure. Okay, whereas endogenous spores such as sporangiospores and zoospores are enclosed in a structure, in a sac, and then they are released outside. So such spores are called as endogenous spores, whereas naked spores which are made outside are called as ectogenous spores. Okay, your conidia is of two types: unbranched as seen in aspergillus, okay, unbranched conidia, and branched conidia as seen in penicillium. Okay, then we have sporangiospores seen in mucor and zoospores seen in Oomycetes. Okay, or Phycomycetes. Yes. So life cycle involving asexual spores. So as we see, conidia, they germinate to form a hyphae, and that hyphae again grows conidia. Conidia is made up of the entire conidia is made up of conidiophore. Okay, whenever there is a phore involving, it means stalk. Okay, so conidia bearing stalk is called as conidiophore. Okay, yes. The second one is sporangiospores. Sporangiospores is nothing but the sporangiospores is released from the sporangium. Okay, and then which germinates and form the mycelium. These mycelium again form fruiting bodies called sporangium, which produces sporangiospores. Very simple. Okay, and the third type is zoospores, which is nothing but the zoospore germinate to form a immature thallus, and that immature thallus forms a mature zoosporangium, which encloses your zoospore, which is again released when the zoosporangium breaks open. Okay. Yes, the third and the final part of reproduction is sexual reproduction. Okay, this happens in three ways: by oospores, ascospores, and basidiospores. Okay, they are produced in distinct structures called as fruiting bodies. So not only sexual spores, any spore forming structure, okay, a structure where spore is produced, such bodies are called as fruiting bodies. Okay, in your higher fungi, you can see the common example is your mushroom. Okay, mushroom is not the main body, but it is the fruiting body of the basidiomycetes. Okay, yes. So the first cycle shows ascomycetes. Ascomycetes as such is means sac fungi. Okay, so the fruiting body is in the shape of a sac. So what happens? The mycelium produces ascogonium. Okay, ascogonium undergoes plasmogamy, which is nothing but fusion of nucleus, and produces a n plus n state, which is nothing but your dikaryophase. Okay, the dikaryophase then fuses to form your fruiting body, immature fruiting body, and then your immature fruiting body undergoes meiosis to form your mature fruiting body, which contains your ascospores. Okay, ascospores, which again get when your ascocarp dehisces, it gets released from the ascocarp, and then it gets released, settles down in a appropriate place where it can germinates and forms the mycelium, and then the cycle repeats. Okay, similar thing for basidiocarp. The mycelium is present, the mycelium is of two strains, strain 1 and strain 2. Okay, it is a positive and negative strain, which is it can also be considered as female strain and male strain. There's no such thing as male and female, but a positive strain and a negative strain can be considered. Okay, they fuse to form again dikaryophase, where the nucleus is not fused, but only the cells are fused. The mycelium is present in n plus n state. Okay, one cell having two nucleus. It's called dikaryotic phase or dikaryophase. Okay. So when this dikaryophase fuses and forms what do you call it, an immature fruiting body, then it matures to form a mature fruiting body called as, here it is called as a mushroom, and it has different structures called as peleus. Okay, the gills, the annulus, and the stalk or the stipe. Okay, so inside the on the undersurface of the mushroom, in the gills, there are structures called as basidium. Okay, which is nothing but your basidiocarp. Okay, basidium on which basidiospores are produced. Okay, these basidiospores, when they are mature, they are released into the atmosphere, settle down on an appropriate surface, and then they germinate into mycelium. Okay, and when the time comes, they again fuse, and then the cycle repeats. Okay, same thing goes for oomycetes, where there are two different kinds of strains. There's an oogonium, which is a female reproductive part, and then the antheridium, which is a male reproductive part. They produce spores which fuse to form a new type of spore, which is called as oospores. Okay, then oospores germinate, and then they form the mycelium. The mycelium grows, and then when it becomes mature, it again produces oogonium and antheridium, and then the cycle progresses so on. Okay, now let us see the 3 steps in sexual reproduction. Okay, there are 3 steps in every sexual cycle. There is plasmogamy, which is the fusion of protoplasm or the fusion of cytoplasm of 2 motile or non-motile gametes. Okay, or 2 motile or non-motile spores. Okay, it is called plasmogamy. Plasma means cytoplasm, plasmogamy. Okay, then the second part is called as karyogamy, or fusion of 2 nuclei. Karyo means nucleus, no? So fusion of nucleus is called karyogamy. Okay, and then we have meiosis in the zygote. After karyogamy, there is immediate meiosis taking place, which results in haploid spores. Okay, so now there is one peculiar thing seen in fungi, where plasmogamy, between plasmogamy and karyogamy, there is no time period. As soon as the plasmogamy takes place, karyogamy also takes place, and you form a zygote immediately. But in fungi, there is a time period between plasmogamy and karyogamy. Once the plasmogamy takes place, the karyogamy does not take place immediately, but it stays in a a dikaryophase, where the cell is fused, but the nucleus from the 2 parents is still there separately, present there separately. They do not fuse. So such a stage is called as dikaryophase or heterokaryotic phase, n plus n phase. Okay, so yes, the hyphae is present. The hyphae produces asexual spores and produces more hyphae. That is one cycle. Okay, it produces asexual spores, and then it produces hyphae. Then the positive and the negative strain of the hyphae fuse to form a heterokaryotic phase. Okay, through plasmogamy. This heterokaryotic phase might be present for a small amount of time or a large amount of time. Okay, we will see the examples in the upcoming slides. Okay, so once the karyogamy is done, once the fusion of nucleus is done, it forms a zygote. Okay, the zygote again stays for a certain amount of time before undergoing meiosis. Once it undergoes meiosis, it becomes haploid, and spores are formed, and those spores are haploid spores. Okay, yes, and those spores again germinate to form haploid hyphae, which again fuse to form a heterokaryotic or dikaryotic hyphae. Okay, so in sexual reproduction, 2 haploid hyphae come together and fuse. Okay, so in some fungi, fusion of 2 haploid hyphae immediately results in diploid cells. Okay, here they are talking about Phycomycetes. Okay, where the 2 haploid hyphae fuse to form a diploid hyphae. Okay, which undergoes meiosis to form haploid spores. Okay, here in the flow chart or in the Venn diagram, we see that there is a life cycle of basidiomycetes. Okay, where your haploid phase is there, and then the majority of the part is your dikaryotic phase. So in basidiomycetes, they have an extended dikaryotic phase. Okay, now dikaryotic phase dominates the diploid phase. Okay, dikaryotic phase dominates the diploid phase. Okay, which is when your mushroom is present. The mushroom is present in the dikaryotic phase. You can see that your hyphae, your positive hyphae and your negative hyphae, they are your nucleus, you can see as red color and blue color are separate, they are not fused. Okay, before undergoing meiosis and forming haploid spores. Okay, so they fuse to form the zygote in the mushroom itself, and they mature as basidium. Okay, and which are released as spores or haploid spores. Okay, the second part is your ascocarp. Okay, ascomycetes. So in ascomycetes and basidiomycetes, dikaryotic stage or dikaryophase is seen as discussed. The dikaryophase is elongated in basidiomycetes, whereas it is short-lived in ascomycetes. As you can see here in the diagram, that the dikaryotic phase is shorter when compared to the basidiomycetes. Basidiomycetes is longer, whereas in ascomycetes it is shorter. Okay, phase is shorter. So yeah, the same thing happens when the ascocarp is formed. The dikaryotic phase is present, where the fertilization takes place or the karyogamy takes place. Karyogamy is fusion of nucleus. Okay, it takes place, and then immediately meiosis takes place, formation of haploid spores are seen of two different strains. It can be a positive strain or a negative strain. Okay, which again germinate separately to form a hyphae. The hyphae mature, form fruiting bodies which have those spores, and then they fuse to form what you call it a dikaryotic phase again, or dikaryotic mycelium. Okay, so later the parental nucleus fuse and the cells are become diploid. Fungi form fruiting bodies where reductional division takes place, resulting in the formation of haploid spores. Okay, so this dikaryotic phase is seen in your fruiting bodies of ascomycetes as well as basidiomycetes. Okay, so when the question comes that what is the ploidy of mushroom, it is dikaryotic phase, n plus n, not 2n, not n, but n plus n. Okay.